It's a very new technique that I didn't really know much about it before, and how I usually describe it is that it's an immunostaining, but with 30 to 35 antibodies at the same time, which is usually quite mind-blowing for people that have always done normal immunofluorescence. And they usually ask me, do you stain them with all of the antibodies at the same time? Like, yes, all of them at the same time. And so how it works is that usually with immunofluorescence, we are limited by the number of antibodies that we can use at the same time because of the signal detection tool, which are usually based on the number of fluorophores that can be detected by a fluorescent microscope. So we usually can use up to 4, maximum 5 antibodies, really. With imaging mass cytometry, we are instead tagging the primary antibodies with metal isotopes. So, which means that we can use as many antibodies as the metals that we have available, and the metals that we're using are lanthanide, which are very rare metals that can be found on Earth. And obviously the limit of the number of metals is just based on nature of how many metals they can be found. So what happens is that after we have tested and optimized the antibody with a normal immunofluorescence, we have a protocol for tagging the primary antibody with the selected metal. And we then stain our tissue with these metal-tagged primary antibodies and then bring it to the Hyperion, which is the equipment that we're using for processing the slides that we stained. And this very complex machine that people also ask me, I mean, I'm not doing the mass spectrometry here, so like, is a machine doing it? So I don't know much about mass spectrometry, but the overall concept is that the machine is able to ablate the tissue using a laser that literally removes pieces of an area of the stained tissue, and this is ablated with an area that is 1 micrometer times 1 micrometer at a time. So this also sets the resolution of the image that is obtained. And each of these tiny pieces are then processed for mass spectrometry, and where basically all of the atoms that are present in this tiny piece of tissue, plus the antibody, plus the metal, are being analyzed. And therefore you would detect, I guess, all of the carbon, hydroxide, oxygen atoms, but then you will also have these rare metals. And because these metals are so rare, it's very unlikely that you will have them in your body. So if the metal is detected, it means that it was there because of the antibody. And then the number of metals that are being detected are equal, let's say, to the number of antibodies that were present, and therefore to the signal. It's translated directly into the signal of that specific piece of tissue. And at the end, the signal from each of these 1 micrometer pieces of tissue are mapped back to the original area that was ablated. So this is how the technique works, and the pros are that you can analyze, you can use many antibodies at the same time, and it's also not, because we're working on metals, we are not, the signal is not confounded by the autofluorescence of the old human brains that is usually brought by lipids accumulation. And there's also very little crosstalks between the different channels. So it's usually, if you get a signal, it's quite very much specific to that antibody, and it's not because of a closed channel that is leaking into the one that you're looking at. And obviously you're looking at protein expression, which is really good. And that's it, I think, for pros. For what concerns the cons, it's a very expensive technique for now, which is what is limiting its usage at the moment. But as it was for transcriptomic analysis, I'm hoping that in the future, with the technology developing further, it will become even more accessible for even labs that don't have the availability of buying such an expensive equipment. Obviously, then antibodies are also quite expensive to order and to optimize, so it's quite a long process to perform. However, I guess that once you have identified the antibodies that are working fine, then you don't really have to go through this optimizing process, and it should make everything a bit shorter. So I guess it's something that it could be easily translatable to a facility in an institute rather to have the single researcher doing it, because it was quite time consuming. The other downside is that we lose, the resolution is not super high, so it's, again, the resolution is based on the size of tissue that the laser can ablate, so it's 1 micrometer resolution. So it's equivalent to a 20x confocal, which is okay, but not great. So obviously, I usually talk about co-expression, but it's actually better to say co-localization of signals. So it's very difficult to say whether that cell is specifically expressing all of the markers that you're seeing, but it's better to say co-localization and then validate your results with an immunofluorescence. And the other negative side is that the way the technique works is that you lose the 3D dimensionality. So while with the confocal, you will have all of the stacks in your tissue, so you will literally be able to see your cell in 3D. In imaging mass cytometry, the whole section of tissues is ablated, so you will have just a max, basically, yeah, all of the signal from the whole section will just be squished in one single stack. So it's not ideal, but we are ablating very thin sections of 8 micrometers, so there is a chance of having cells that are overlapping, but it's very little.